So I'll introduce you. So this is Professor Damien Arrigan. So Damien is my PhD supervisor, former, I guess, PhD supervisor. And yeah, so Damien began, he spent time in the UK, in Southampton and Manchester, and then he went to Cork, and that's where I worked with him in, it's now called the Tyndall National Institute. And then he left for Australia just around the time I was finishing my PhD, and he's been there since. So that's like more than 10 years at this stage. So yeah, Damien, looking forward to your talk today, and best of luck.
Thank you very much. Okay, thanks, Mihal. Good evening, everybody. It's 7 p.m. here in Perth, Western Australia. So good evening, good afternoon, good morning, wherever you are around the world. Thank you to Mihal for organizing this excellent series of seminars. I've really enjoyed all the ones that I've listened to so far. I think it's a great initiative, and well done to Mihal for doing that. I'd like to talk to you today about some of the work that was done here during a visit we had last year from Benjamin Viada, who visited our lab from the Universidad Nacional de Cordoba in Argentina. He came here for a 3-month visit to look at some electroanalytical chemistry, and he did some great work on the detection of perfluorooctane sulfonate. So I'm going to talk to you about this work using ion transfer voltammetry to detect this important environmental contaminant. So as I said, the work was done by Benjamin Viada, or Benja as we know him. He came here from Cordoba in Argentina, and I believe he traveled westward out of Argentina and went all the way around the world via Sydney, ended up in Perth here on the west coast of Australia. And he spent 3 months here working on the electrochemical behavior and detection of perfluorooctane sulfonate, and eventually used it in his PhD thesis back home in Argentina. And he's now finished, and he's now Dr. Viada. So congratulations to Benja on successful completion of his PhD. Per and polyfluoroalkyl substances, or PFAS, as they're commonly referred to, are a worldwide pollution problem. They're persistent environmental contaminants. They're highly stable. They exhibit bioaccumulation effects, and they have toxicity effects as well in animals and plants and other living things as well, I guess. They've been widely used around the world for many industrial and commercial applications. Some examples are listed there on the screen. So they've been used in paper and packaging, on stain and water resistant coatings, even in non-stick cookware, which we all use perhaps. In industrial applications, probably the biggest use has been in fire resistant foams. So they've been widely used in, around airports and around defense bases worldwide. And as a result of this wide use around the world, there's contamination everywhere with these PFAS substances. So we list here on the bottom here just 2 examples of PFAS substances, perfluorooctanoic acid and perfluorooctane sulfonate. These are the 2 most common PFAS substances, and they're the most common contaminants in the environment, and also in human samples that have been analyzed. So these are surfactant materials. They've got a long fluorocarbon chain and an ionizable or ionized head group. So they exhibit surfactant properties, and this is the basis of much of their applications around the world. So because of their bioaccumulation and toxicity effects, there's a lot of interest worldwide in monitoring their behavior. This is just some examples of some pictures showing their use in firefighting foams, for example. So even in firefighter training bases, there's lots of contamination of the environment around these bases. That contamination gets into the ground around the bases, can even seep into the groundwater. So the water that is extracted then to fabricate or to manufacture drinking water can already be contaminated with these types of substances. So there's a lot of bioaccumulation of these materials, toxicity effects, various biological effects of these materials. So as a result, there's a need for methods for their analysis. Here's a map of the PFAS contamination problem around Australia. So we're here in Perth. So wherever there's one of these symbols, it indicates a site where there's some PFAS contamination. So around Perth, there's plenty of sites. Around southeastern Australia, there's lots of contamination of the groundwater and the soil with these materials. And this PFAS contamination actually tracks very well with the human population all around Australia. So wherever there's a a high density of humans, there's also a high contamination of the local environments with these materials. How do we measure PFAS so that we can assess the environmental contamination? Well, like many things, combinations of chromatography and mass spectrometry are widely used. So typically liquid chromatography, mass spectrometry in one form or another, or gas chromatography, mass spectrometry in one form or another are used for measurement of these types of substances. If you want to go on site and measure these substances in a rapid fashion, LCMS and GCMS are not very amenable to producing fast results. So the traditional approach would be to go onto a site, take samples and send them to a remote laboratory and wait maybe 24 or 48 hours or even longer to get your detailed analysis back. So if you want to do rapid on-site analysis, maybe to monitor remediation processes that are being used to try and clean up contaminated sites, these traditional LCMS and GCMS methods are not suitable. In the last probably 5 or 6 years, there's been lots of interest in developing sensors for these compounds. And I've listed some electrochemical approaches here, various types of electrochemical approaches using molecularly imprinted polymers or metal organic frameworks or bubble nucleation methods to detect picomolar concentrations of different PFAS materials. So with these electrochemical approaches, people have been able to get down to quite low concentrations. And these are the concentrations that are required to, if you want to be able to monitor the environmental concentrations of these substances. The bit that piqued my interest a few years ago, or the paper that piqued my interest a few years ago on this topic, was from Garada et al., which is Shigeru Amemiya's laboratory in Pittsburgh. They published a paper in 2014 on the detection of PFOS at 50 picomolar limits of detection based on ion transfer across a water organogel interface. So this is like a liquid-liquid interface where we have the oil phase or the organic phase is gelled with PVC to make it more mechanically stable. And using a stripping voltammetry approach with a 30-minute pre-concentration time and a rotating electrode to enhance the mass transport, they're able to detect these picomolar levels. So we're particularly interested in seeing, could we develop this idea of liquid-liquid electrochemical detection of PFOS and use that as a basis for detection in samples in Australia. So for some years now, I've been working on electrochemistry at the interface between two immiscible electrolyte solutions with various collaborators. I actually started all of my work on electrochemistry at the ITIES in collaboration with Robert Dreif, probably about 20 years ago or so, more than 20 years ago, when I was working in Manchester, and we had some discussions with the local electrochemistry community, and that led to Rob and myself collaborating on exploring analytical aspects of electrochemistry at liquid-liquid interfaces. So what we're interested in doing in my group is looking how we can explore or harness the transfer of ions across a liquid-liquid interface formed between water and an organic phase and use that as a basis for detection. So rather than looking at oxidation or reduction processes at the surface of an electrode, we're looking at ion transfer processes across a liquid-liquid interface. So in this case, we're looking at, say, for example, simple ion transfer reactions where an ion is transferred from the water phase into an organic phase, and that transfer of ions across the interface gives us a current, which we can measure. We can also look at facilitated ion transfer reactions where we have quite a hydrophilic ion, which doesn't transfer very easily across the interface. We can lower the energy requirement to transfer that ion across the interface by adding a ligand or an ionophore to the organic phase. So we're interested in using these types of facilitated ion transfer reactions or simple ion transfer reactions implemented in a liquid-liquid interface electrochemical cell as a basis for chemical sensing or chemical detection. Of course, these traditional liquid-liquid electrochemical cells are not very useful when it comes to sensing. You couldn't imagine picking up this cell and walking out onto a polluted site and trying to do contaminant analysis on site with this sort of mechanically unstable interface. So we've also got a limitation when it comes to analytical performance with these large liquid-liquid interfaces. So when I say large, that might be a half a centimeter or a centimeter in diameter across there. With these large liquid-liquid interfaces, the analytical sensitivity is limited to about micromolar levels. And for environmental monitoring applications and even for lots of biomedical applications, we need to get down to sub-nanomolar concentrations even to have a hope of being able to detect anything usefully. So Benja was interested in coming to our lab to see how he could use electrochemistry at liquid-liquid interfaces. He already had experience in using electrochemistry at liquid-liquid interfaces to characterize PFAS substances. So he'd looked at using cyclic voltammetry to measure the critical mycel concentration of a range of PFAS substances. So these are the values he got with cyclic voltammetry at liquid-liquid interfaces compared to traditional methods applied also at liquid-liquid interfaces. So we wanted to build on Benja's experience in electrochemistry with PFAS at liquid-liquid interfaces, but also combine it with our approach to miniaturizing liquid-liquid interfaces and to try and enhance the analytical performance that we could get. So we've been working quite a lot on miniaturized liquid-liquid interfaces, where the idea is to make the interfaces small, either micron size or even nanometer sized interfaces, with the idea of enhancing the mass transport, reducing the ohmic resistance, and overall trying to improve the signal to noise ratio. So of course, many people before us have been looking at miniaturized interfaces, going back to Hubert Giraud's work in the 1980s on using micropipettes to make miniaturized interfaces. Subsequent work by Xiao and Merkin on looking at nanopipettes. And of course, Robert Dreif, who introduced the idea of using nanofiltration membranes to make arrays of nanoscale interfaces. For anyone who is interested in the background to miniaturized ITIES, there's an excellent review by Yuhan Xiao in Chemical Society Reviews, published in 2011, and I'd recommend everyone to read that if you want a thorough background on the electrochemistry of miniaturized ITIES. These are just some examples from the literature of miniaturized ITIES. So this is a pipette, a micropipette, or maybe a nanopipette. And the idea is that you have, say, the water phase inside the pipette, the organic phase outside the pipette, and at the mouth of the pipette, you get the liquid-liquid interface formed, and we get ion transfer across that interface that we can use to measure. In their case, they're measuring the kinetics of ion transfer, but we would be interested in using that ion transfer as the sensor signal, for example, and developing an amperometric signal. You can imagine that the current at a single micropipette or nanopipette is quite small. So arrays of micro or nano interfaces could also be used. Here's an example of arrays of nano holes that are formed in polymer membranes by Hubert Giraud's group using laser ablation. And you can use this to make an array of nano or an array of micro liquid-liquid interfaces, and then use that for detection of nitrate or phosphate or other environmental nutrients, for example. So our approach to miniaturization, or one of our approaches to miniaturization of liquid-liquid interfaces, was to use laser ablation of glass membranes. And the idea here is that we use a laser to basically punch holes in a glass membrane. And this glass membrane is just a simple microscope cover slide that we buy from regular laboratory suppliers. So it's a glass membrane. It's about 300 micrometers thick, and we can punch holes in it using this laser. And of course, the laser ablation process gives us these cone-shaped pores. So we've got a wider opening where the laser hits the glass, and a narrower opening where the laser exits through the glass. And this video up here just shows the formation of individual holes in the glass membrane using the laser. It's quite a fast process to make individual holes, and we can make arrays of holes quite easily by just rastering the laser across the surface of the glass and punching each hole in the glass one at a time. With this laser ablation process, we can make holes in the glass membrane that are about 50 microns in diameter at the laser entry side, and about 20 microns in diameter at the laser exit side. So as I said, we get these cone-shaped micropores in the glass membrane, and the idea is that we can place these glass membranes, these Holy glass membranes, at the liquid-liquid interface. Ideally, one of the phases will fill the pores in the membrane, so we get an interface formed either at this opening or at this opening, depending on the configuration of the membrane that we use in the experiment. So here's some electrochemical behavior of ion transfer electrochemistry at these glass membranes formed by laser ablation. And you can see with this simple cyclic voltammetry characterization just for tetrapropylammonium ion transfer across the water DCE or water DCH interface or array of micro interfaces, we get a cyclic voltammogram that indicates we're getting radial diffusion on the forward scan, which is consistent, or we get a steady state current on the forward scan consistent with radial diffusion from the aqueous phase towards the interface. and on the reverse we get a peak-shaped response in the voltammogram consistent with linear diffusion within the pores. You can see we get quite a different shape in the voltammogram depending on whether we have this inverted cone shape with the wider opening open to the aqueous phase, or with the pores turned around where we have the narrower opening open to the aqueous phase. So what we think is going on here, we get the enhanced response here on the reverse scan of this voltammogram, because during the forward scan, ions transfer into the pore, and because of the nature of the pore wall, diffusion out of the pore into the bulk of the organic phase is hindered, so we get a buildup of concentration within the pore, and so as a result on the reverse scan, back down to lower potentials, we have a larger effective concentration of the analyte in the pore, so we get a larger peak current as a result. So in our electrochemical experiments with PFOS, or perfluorooctane sulfonate, this is the typical electrochemical cell we've used here. So the aqueous phase here would typically be 10 millimolar lithium chloride spiked with different concentrations of PFOS. And other than that, we've got our regular background electrolyte in the organic phase. So that background electrolyte is an organic salt that's made up of an organic cation and an organic anion, and they're happy to stay in the organic phase. They don't transfer from the organic phase into the aqueous phase unless we apply sufficiently large potentials. We use a simple 2-electrode electrochemical cell. This is our glass membrane perforated by laser ablation. We've got the organic phase inside a glass tube onto which that glass membrane is glued. The organic phase fills the holes in the membrane to form interfaces like this or like this. So for the PFOS experiments, we've used exclusively this configuration of the glass membrane. Sorry, we've used this configuration of the glass membrane. So we have the wide opening exposed to the aqueous phase. We have an inverted cone-shaped pores in the membrane, and when the ions transfer from the aqueous phase into the organic phase, they get trapped in the pore, so the concentration here builds up during that scan and gives us these large peaks on the reverse scan of the cyclic voltammogram. So here's a typical cyclic voltammogram for the detection of PFOS. So the black line here is just the background scan. So if we scan down to sufficiently low potentials, we eventually get to a potential where we transfer background electrolytes, and that limits our available potential window. If we transfer to sufficiently high potentials on the forward scan, we would see a similar sort of increase in current due to background electrolyte transfer. That's in the blank, where the concentration of PFOS is 0. When we spike PFOS into the aqueous phase, in this case, we scan down to lower potentials because we're dealing with an anion. As we transfer the anion, in this case, from the aqueous phase to the organic phase, we get a negative current. We continue down to more negative potentials. We'll eventually get background electrolyte transfer as well. We transfer back up, and we get a reverse transfer of the PFOS from the organic phase back into the aqueous phase. So the forward scan is transfer of PFOS from aqueous into organic phase, and the reverse scan is transfer of PFOS from organic phase back into aqueous phase. So it's not an ideal response. So there may be kinetic effects in the ion transfer process, or there may be some uncompensated resistance in the electrochemical cell, which we haven't worried about. So it's not an ideal response, but qualitatively it does tell us that PFOS can be detected. So we can detect it by ion transfer from the aqueous phase to the organic phase, or we can detect it by ion transfer from the organic back into the aqueous phase. So if we're interested primarily in analytical chemistry and how we can use this form of electrochemistry to detect low concentrations of this substance, maybe a more useful technique to use is differential pulse voltammetry. So here we've got 2 sets of differential pulse voltammograms. In this case, we call this a forward scan differential pulse voltammogram, or set of voltammograms where we're scanning from a high potential to a low potential. As we scan down, the PFOS transfers from the aqueous phase into the organic phase and gives us these peak currents which are proportional to the concentration. Or we can do the opposite. We can start at a potential more negative than the transfer potential, and we can scan up in potential. So in this case, when at the initial potential here, we're applying, or at that initial potential, the PFOS is spontaneously transferring from the aqueous phase to the organic phase. And then as we scan up, we're transferring it from the organic phase back into the aqueous phase and getting these peaks, which are proportional again to the concentration of PFOS in the aqueous phase. So a step further from this is to use a method called differential pulse stripping voltammetry. So with these scans here, we're applying the initial potential, but we're not waiting at any time here. We're just immediately scanning towards positive potentials and removing the PFOS from the organic phase back into the aqueous phase. With differential pulse stripping voltammetry, if we apply this initial potential, then wait for a set time, so we're maybe waiting for 30 seconds or 60 seconds or 120 seconds or whatever, that would give us a pre-concentration effect, which allows us to extract the analyte from the aqueous phase into the organic phase for that set time. So we build up the concentration in the organic phase, trapped in the pores even further, and therefore we should get a higher peak current as we scan to higher potentials. So this method of stripping voltammetry is well known for giving improved sensitivity and for lowering limits of detection. So if we do that approach here, and what we're doing is we're applying an initial potential of 0.1 volt. We're holding that potential for a set time, and then we're scanning to higher potentials to extract the PFOS from the organic phase back into the aqueous phase. So as we increase that pre-concentration time, waiting at this initial potential for different times ranging from 0.5 minutes up to 30 minutes, we can see that the peak height increases and we get an improvement in the stripping peak current with pre-concentration time as a result. So we're working at fixed pre-concentration potential that extracts the PFOS from the aqueous phase into the organic phase. After a fixed time, we then scan to higher potentials and strip the PFOS from the organic phase back into the aqueous phase to give us our analytical signal. The other important point to note here is that we're working in unstirred solution, so neither are we stirring the solution nor are we rotating the electrochemical cell or rotating the electrode to enhance the mass transport. So we're relying on the radial diffusion that occurs at micro liquid-liquid interfaces to give us our enhanced mass transport and allow us to work in unstirred solution. So for subsequent work, we've chosen our 0.1 volt pre-concentration potential and a 5-minute pre-concentration time, which should be that peak there. And we've looked at how the concentration effect, how the concentration impacts on the stripping current that we then measure subsequently. So here we have the effect of concentration. So different concentrations in the range between 0.1 nanomolar and 1 nanomolar PFOS. So we apply our 0.1 volt pre-concentration potential, we hold it there for 5 minutes, and then we scan to higher potentials and strip the PFOS from the organic phase out into the aqueous phase. And that stripping current gives us, we get a peak current that's proportional to the concentration over that range that we studied. And looking at the linear regression analysis of this, we can get things like the standard deviation of the slope, we can get the intercept and the standard deviation of the intercept, and a well-known parameter in analytical chemistry is the limit of detection, and that's a statistical definition based on, typically it's 3 times the standard deviation, or a signal that's 3 times the standard deviation of the blank larger than the blank. So if we take the intercept here as our blank signal and get the standard deviation of that blank from the statistical analysis, we can get our standard deviation, we can calculate our limit of detection based on 3 times the standard deviation divided by the slope, and we get 0.03 nanomolar as our limit of detection. Or if we convert that to mass per volume concentration, it's 0.015 micrograms per liter or 0.015 parts per billion. So we're at the sub parts per billion detection level for this PFOS contaminant. And if we can compare that to drinking water limits set, for example, in the US by the Environmental Protection Agency or in Australia by the Department of Health, we can see our calculated detection limits are below the limits set for drinking water contamination levels for this contaminant in both the US and in Australia. So it looks like, based on this performance, that we can detect these substances, or we've got a good enough detection capability to be able to detect these substances in drinking water at least. Of course, all of this is in pure electrolyte solution. This is in 10 millimolar lithium chloride aqueous phase. What happens in real samples? Real samples are much more complex than 10 millimolar lithium chloride. Real samples could be something as complex as blood. These PFAS materials have been found in human blood samples. So could we eventually think about trying to analyze human blood samples using this form of electrochemistry? If we want to be able to do that, then we need to consider what happens in real samples. So Benja's experiments in real samples consisted of examining a range of different types of waters. So drinking water just from a drinking fountain in the building, tap water just from a tap in the laboratory, and seawater that he took from one of his many visits to the beautiful beaches all along the west coast of Western Australia. This is Cottesloe Beach, and the ocean there is the Indian Ocean. Lots of beautiful beaches all along this. So whenever any of you get a chance to visit Perth in the future, when all this COVID pandemic is over, then be sure to visit some of our beautiful beaches. We didn't find any peaks for PFOS in any of the samples we analyzed. So perhaps if there is contamination in any of these samples, they're below the detection limits of our method. So what we did was we spiked these samples with different concentrations of PFOS, again over the range from 0.1 to 1 nanomolar PFOS. In drinking water, we get these beautiful peaks, maybe indicating the purity of the drinking water here in Perth. In laboratory tap water, we get nice peaks as well. There's this unexplained shoulder here as well. And in seawater, we can also get some very nice peaks for detection of spiked PFOS in the seawater. We also have this increase in current after the PFOS transfer current, which we haven't been able to explain, but it also happens in synthetic seawater. So if we make up synthetic seawater using well-known recipes, we also get these increases in currents that we don't see in, say, a pure natural water sample. So we can look at the sensitivities and detection limits for all of these different types of water samples, and this table here summarizes what happens with regard to our detection limit. So our pure water sample, ultra pure water there is the our pure electrolyte solution. That gives us our lowest detection limit, 0.03 nanomolar or 30 picomolar. It also gives us our highest sensitivity, our calibration graph slope. As we move to more complex samples, we get a loss of sensitivity, so the calibration graph slope decreases. We also get higher calculated limits of detection, so there is a matrix effect. So it means when it comes to analyzing real samples with this approach, we would have to take that matrix effect into account by how we calibrate. We couldn't just use simple calibration graphs. We would have to use something like standard addition methods to overcome this matrix effect. So despite that loss of sensitivity and increase in limits of detection as we move to more complex matrices, we can still see that with a micro ITIES array with 5 minutes pre-concentration with differential pulse voltammetry and unstirred solution, we're still getting quite low detection limits. And there is, of course, scope to improve the detection limits and to improve the analytical performance by moving to longer pre-concentration times, or maybe to think about other ways to enhance the mass transport that we use during the pre-concentration step, because that really is the crucial way of controlling the sensitivity of these stripping voltammetry approaches. So how am I doing for time? So it's about half an hour now, so I'm reached my conclusions, Mihal. I hope that's okay. The conclusions are we can make wholly glass membranes by laser ablation, very simple process. We don't do it here. Actually, it's done by a service laboratory in other parts of Australia. We can we can achieve the detection of perfluorooctane sulfonate, a typical PFAS substance, by ion transfer voltammetry at arrays of micro liquid-liquid interfaces formed in these glass membranes. Using a short pre-concentration time and unstirred solution, we can get down to concentrations lower than the regulatory limits set for drinking water in some countries around the world. But the important point to note really is that these are very much preliminary results. There's still lots to do. Can we detect other PFAS substances? What happens if we have mixtures of PFAS substances in samples? Can we improve the detection limit down to lower concentrations? Can we get a better handle on the matrix effects? And can we start to analyze samples from actually known contaminated sites? And finally, can we develop all of this into a robust device that could be used by someone who's not an expert analytical chemist or electrochemist, or someone who's not even trained in science or technology, that they could use it in a sufficient way to provide some information about drinking water in their own home, for example. So with that, I'll just thank Benja and his supervisor, Lydia Yudi, for their collaboration on this topic. I thank members of the Electrochemistry and Sensors group here at Curtin University for all of their ongoing great work on exploring the electroanalytical opportunities of electrochemistry at liquid-liquid interfaces, and in particular, the Australian National Fabrication Facility the Optofab at Macquarie University, and the node at the University of South Australia, where the fabrication work is done. And finally, just a special thanks again to Mihal for organizing this series and for all the great work you do in electrochemistry. So as Mihal said in the introduction, he was one of my PhD students in Cork many years ago. Looking at this picture, I think he started his PhD when he was about 12. Okay, thanks everybody.
Thanks, Damien. That picture is going to haunt me like forever.
It is, yeah. It'll be coming back forever.
Yeah. I don't think you've ever given a presentation without showing that picture, actually.
I have many times. You just haven't seen them.
Yeah, maybe. Okay, so if you could stop sharing your screen, and we'll see if anyone